Assalamu alaikum wa rahmatullahi wa barakatuh. Welcome all dear students. And today's lecture is going to be about commensals and pathogenic microbiota of human body. This is me, Dr. Zaim Al-Jabri, and I'm going to go through the following objectives. So in this lecture, we are going to cover the following objectives. It's very important for us to know as this very first beginning of this course that there is very clear distinction between what we call normal flora, which is the good bacteria that we have, and also the microbes that are pathogenic, and what is in between, what we call the symbiosis or symbiotic microbes, which is actually what causes the different microbes to live in the same environment in a symbiotic relationship. So now we are going to define in the next slide what do we mean by normal microbiota and why is it so important for us. So when we talk about microbiota or we mention the term microbiome, it's actually two different words for almost the same thing, but there are some differences. So before that, we need to know that our human body has what we call commensals. So when we say commensals, it's basically the same as normal flora. When we say commensal microbiota or only microbiota, actually this is what we refer to also as microbiome, is the good bacteria, and it is located in different parts of our bodies as a collection of bacteria, and we call them community. And it's very, very important for us to know in this course that before we think of microbes as enemies and something that we have to fight, we have to think of them actually as friends. So how many of these good friends we have on our bodies? Actually, there are so many. In fact, it's believed that human body is built up by almost 10 trillion cells, but on the other hand, we have 100 trillion non-pathogenic bacteria or microorganisms that is present on our bodies or inside our human bodies. And these billions, trillions, or millions of microbes actually are present on a large surface area on the different parts of our bodies, including the skin, our upper respiratory tract, our gut or colon, and so on. So it's very important for us to remember that microbiome also is a term that is becoming very popular nowadays, and it's very important also for us to know. And it refers to the collective genomes of all microorganisms. So it's not just bacteria, it's actually believed to be bacteria, fungi, viruses, as well as protozoa. They all reside within one person and they share all that body space. So it's very important for us to remember these very important facts about these microbes or microbiome. And I want you also to remember that the difference between microbiome or microbiota is that microbiome is an older term and it's used to refer to these good bacteria or good microbes on our bodies. But now microbiome is a more general and it's actually a newer term, which basically collectively means the genomes. So what is the difference between a bacteria and its genome? Is actually because some of these microbes actually exchange their genomes or genetic material. So it's actually not just the physical presence of these microbes, but also the genetic material of these microbes that also determines a large part of this microbiome. So now let's move on to the next slide to define few terms related to the microbiome. So actually, in our bodies, we have different communities of microbiomes, as we have learned earlier. And we have three different terms. The first one refers to microbial community that are variable at different body sites but on the same person, and this is what we call different ecosystem. So what do we mean by that? This basically means that one person has a community of microbes that live in his, let's say, mouth that is different from the community of microbiome that lives on the skin. Although there might be some similarities, but they are different communities or different ecosystems. And that will also lead us to the second term, which is the what we call common ecosystem. So this refers to the bacteria of a specific site of the human body, but in different persons, they always have or almost always have something in common or some common characteristics. For example, if one person has certain type of bacteria, let's say person A have some common bacteria in the mouth or oral cavity, so the other person or person B is likely to have similar microbiome composition in the mouth as well, because bacteria or microbes in general, they have preferable body sites. So if we take different samples, but from the mouth of different persons, we are very likely to see that they are similar. And that's what we are going to learn in the practical session of the normal flora. And this also leads us to the third term, which is the microbiome, rearrangement of the ecosystem when actually a healthy person microbiome looks different from a diseased person due to the changes in the relationship between the different microbiome systems or communities. So these are very important terms for you to remember. Now let us move to the second slide to learn about the favorite places for these microbes to live in our bodies. So what are the favorite body sites, or what we call them hotspots for these microbial communities to live? They actually like to reside at certain individuals, or some of them actually have been there on our bodies since we were born. So that's what we call them native. So they are found on our bodies since we were born, and this is what we call them resident flora. And some of the microbes can also be introduced to your body suddenly when they have not been there, but you have acquired them through, let's say, from being coming into contact with these microbes in the environment or from another person. So this is what is called transient flora that has not been there, but now you have acquired them. So there is a very important and a delicate balance between the microbiome environments that need to be maintained in order to keep our bodies healthy. And any compromise in our immune system, like it's becoming weak, this will actually cause this balance to disappear or actually to become a little bit disturbed. And then this will allow the microbiomes, let's say, which were not very harmful into their first sight, to be now becoming out of control, and now they start going to the wrong place and cause diseases. And this is what we are going to learn more in the tutorial. So let's move on now to the next slide. So students, I hope you have heard about what is called as the Human Genome Project, in which scientists for many years have been trying to sequence our genome. But now there is a huge interest also in understanding the human microbiome, and a lot of information has been acquired from that human microbiome that is very much believed that it's our other genome. So from that we can understand a lot of information, and it's very useful microbiome system we have, because without that microbiome in our gut, for example, we won't be able to digest the food that we eat. So for example, This microbiome in our intestines actually produce hydrolytic enzymes, and these hydrolytic enzymes, without them, they cannot break down our food. The other very important function is the synthesis of so many vitamins, including K, B2, B12, and folic acid. And also, sometimes there's what we call competitive inhibition, in which these good bacteria compete for nutrients to be able to survive in our gut. And in order to do that, this will be helpful because once they take out this nutrition, it will allow it to grow. But on the other hand, a lot of harmful bacteria will die on the way. And also, it's very much now well established that there is a strong connection between the skin and the gut microbiome. And through that, we modulate the innate and adaptive immunity that you will learn into greater details when you learn immunology in this course. And again, it prevents growth of pathogens. And not only that, actually, we have a lot of good bacteria in the skin that produce antimicrobial peptides or substances that will kill the harmful organisms. And also, again, through the competition of nutrients, that balance is maintained so that the good bacteria will outnumber the bad bacteria. So it's very, very, very much important for you to understand that these microbes are not harmful germs like you have learned about the germ theory. In fact, these are good microbes that very much beneficial. Like when you eat something, let's say vegetables, they out of that vegetables you won't be able to get energy without the metabolism that is initiated by these microbiome or microbes or good bacteria in the gut. Now let us move to the next slide. So in this slide, I want you to understand very, very important concept. And actually, it's one of my favorite theories that is emerging and trending. And I'm trying myself to understand it and read a lot about it, which is the interaction between what we eat or the diet that we eat and the microbes that live within us, and how the interaction between these two circles with the genetic materials within our bodies, or the chromosomes, or the expression of our genes. And although this is a very, very hard question to answer at this stage, we need to understand that these three circles interact with each other without being able to actually pinpoint where does this relationship starts. But what we can say here is that we have what we call homeostasis, or a very stable relationship in which the diet that we eat interacts with the bacteria that we have, or in other terms, feeds the bacteria that we have, and then whatever we eat, whatever microbes we have in our bodies, will actually determine which genetic material is going to be expressed or which genes to be expressed. And if we know which genes are expressed, then that would determine whether we are healthy. So we have good, healthy genes. And this will tell us that we have very low risk of developing diseases like allergies, infectious diseases, or resistance to certain infections. And also, if this line is crossed between this homeostasis due to, let's say, disturbance in this microbiome, this what causes actually disease. And the disease condition will be provoked in terms of allergies, metabolic syndrome, obesity, and infections. We will come to that again one more time in this lecture later. So what is a healthy microbiome? We don't know, because there is not one size that fits all. It's actually every single individual of us has a unique set of microbes or microbiome, and so we cannot really define what is good and what is bad. Now let us move to the next slide to understand more about these microbiomes. So now let's go through what are the sterile body sites. When we say sterile, we mean that we do not expect to see or grow any microbes in these places in any healthy individual. And the first one is blood. So blood should always be sterile. Also, brain should be sterile. Bladder should be sterile, otherwise you will end up having urinary tract infection or cystitis. And generally, any internal organ normally should be sterile, like the lung, liver, and spleen, apart from the gastrointestinal tract. And in order to be able to grow these microbes that we isolate from these body sites, we have to actually grow them on an enriched culture in which we use an agar medium which contains a lot of nutrients. And good examples of enriched media is blood and chocolate agars. And on top of that, there are also some body cavities that should be sterile, like the pleural cavity, pericardial cavity, also the abdominal and pelvic cavities. So these are the spaces in between the organs that should always be sterile. Now let us move on to the organs that harbor these good microbes. So the first part that we need to talk about when we are addressing the microbiome is the skin, because it's actually one of the largest organs that we have in our bodies. The surface area of the skin is very large, and that's why we can find trillions and trillions of microbial communities on the skin. And because there are different skin types like oily, dry, or moist, so microbial communities differ between different body sites. Some of them prefer one site over another, and they are not always on the surface. We know that bacteria can prefer to stay on the surface, but actually, when we wash our hands with soap and water, most of these surface bacteria will be killed. And where do we actually compensate for these? The killed microbes will be compensated from the hidden microbial communities that hide within the sebaceous glands and sweat glands and hair follicles. So once we wash our hands, all of these will be uh, outgrowing from these glands to the surface. And if you go back to the anatomy of the skin, the top layer is known as stratum corneum, and that's where most of the bacteria is found. And most of the bacteria actually make or synthesize or secrete what is known as antimicrobial peptides. And why is this substance important? Because we want to keep homeostasis, as we mentioned earlier. So these are released by the bacteria, and this will keep the community of harmful microbes under check. And also because we sweat a lot, this acidic sweat will be acting as defense mechanisms against these harmful microbes. So as I said, a lot of hidden bacteria will be there in the hair follicles, and this will act as the reservoir for compensating for the bacteria on the superficial skin after washing. Now let us move on to name a few bacteria. I know now we start with the heavy stuff, and actually now going to the depth of microbial communities. So when we talk about normal flora of the skin, we have to really think about this. Where did we get this normal flora to start with? Are we born sterile or not? Actually, it's a question that scientists have been trying to address in so many studies, and they believe that once we are born, we are not sterile anymore. We actually get establishing these commensal communities the moments we are born. And actually, the most important microbes that we have on the skin is staphylococci, that you are going to learn in the coming lecture. These are gram-positive bacteria. Second most common is micrococci, and third is anaerobes, which are the bacteria that do not require oxygen to grow. But please remember that on each organ we have these two teams. One team of commensals, the good bacteria, and these pathogens that will always be there in lesser percentage, but in any case that our immunity goes down, these pathogens will outgrow. So the commensals will keep the pathogens in lower numbers. And the most important pathogens of the skin is Streptococcus pyogenes, again a gram-positive bacteria, and another important one is Staphylococcus aureus. So if you want to remember them, it's actually the largest groups of gram-positive bacteria. We call them staph and strep. Now, in order to grow these, we use media called like agar that we will learn into details in the coming lecture. Now let us move on to the second organ. So now we'll move on to the upper respiratory tract, and as you can see from this picture here, what we mean by upper respiratory tract is starting from the nose, so the nasal cavity, and then we go down posteriorly to the pharynx, and then to the epiglottis and the upper part of the trachea. So, what keeps this environment healthy? Okay, so in this slide, I want you to remember before going into details of these microbiome here, we have to remember that our nasal cavity has a primary defense mechanism. And this because we inhale a lot of pathogens through breathing, and we also inhale a lot of dust and a lot of harmful chemicals. So what keeps the nasal cavity clean is the actions of the cilia and also the mucus lining that produces mucus to trap any inhaled microbes. And also there is in the interior lining of the nasal cavity, we have the mucus secreting glands that also secretes a lot of antimicrobial peptides. The most important bacteria that is found in our nasal cavity is called Staphylococcus epidermidis. It's a gram-positive bacteria that is commonly found in almost every individual, every one of us. And this bacteria is known to form biofilms. What we mean by biofilms is a group of microbes that gather and they are surrounded by a layer of a thick matrix of polysaccharides. And there is also another important bacteria that is present, which is called Staphylococcus aureus. This keeps Staphylococcus, actually, epidermidis will keep Staphylococcus aureus in much lesser amount because it secretes protease enzyme that will degrade the Staph aureus. Because as I said, we need these commensals and also we have to a lesser extent pathogens. So the commensal here is the Staph epidermidis and the pathogen is Staph aureus. So it's okay if we have Staph aureus, but if I screen the whole class, maybe we will not find all of us have it, but if I screen you will all have Staph epidermidis. So that's the difference between the two. And if any disturbances happened here, we will see that Staph aureus starts becoming larger in amount and then it becomes pathogenic, which causes infection. Now let us move to the second part of the nasal. After the nasal cavity, we'll cover the oral cavity. So what are the most common defense mechanisms that we have in the mouth? So if you see here in the mouth, we have to start with microbes that are in the saliva, in the gum, teeth, and also biofilms. So again here the bacteria will gather in groups and they form around a matrix. So there's what we call the exopolysaccharide layers that will gather these bacteria, and once it becomes trapped within this matrix, it becomes very difficult to get rid of these bacteria. So what kind of defense mechanism we have in the oral cavity? We have saliva. So the saliva will have a lot of degradation enzymes. We have also a certain amount of low pH that will keep the harmful microbes under check. You have the relatively higher temperature in the oral cavity that again will keep the bacteria under control. There's also the immune system that has a lot of recruitment of different types of antibodies that you also learn later that prevents many species of bacteria from growing and surviving. So, what are the most common microbes that we have in our uh, mouth? Again, it's similar to the previous ones, but before that, we need to remember that these are found there because it's very important. They maintain homeostasis. What do you mean by homeostasis again? Balance. So it's balance that is kept by this oral microflora. It prevents a lot of oral cavity diseases, and also it prevents the spread of these bacteria or protects the spread of the harmful bacteria from going through the oral cavity into our internal organs. So in the oral cavity, we have these microbes that are found in the gum. And what happens is that when we brush or floss our teeth, we are we are cleaning our mouths, but again, we are doing some harm there because if we are doing a lot of harmful or rough brushing, what you do here is that you stimulate a lot of entry of the harmful bacteria to inside the gum, and then it can go from the gum into the blood, causing what we call bacteremia. And this is actually becomes very important in a group of people with heart conditions. So let's say, for example, you want to remove a bad tooth and you went to the dentist. And if you have a heart condition, let's say a condition called congenital heart disease or a heart disease that you had since birth, and this group of people actually dental extraction, although it's a very simple procedure, it becomes very harmful because what happens here is that the harmful bacteria will go through this extracted tooth to the blood, and then it immediately goes to the heart valves. And what happens is that these heart valves, because they are not normal, they are either having irregularities or abnormalities, the accumulation of the bacteria in the heart valve will cause subacute infective endocarditis or subacute bacterial endocarditis. So it's very much known now. There is a big amount of research and evidence that connects the health of our teeth with the health of our heart. So if you have a disease condition in the heart, you are more likely to be prescribed antibiotics before dental extraction so that it becomes a prophylaxis against acquiring any condition in the heart, and so that will prevent the person from having an infection in the heart. So now let's move on to the normal flora of the upper respiratory tract. So when we talk about the upper respiratory tract, as I mentioned before, the most important anatomical site is the mouth. Is actually part of it, oropharynx, nasopharynx, and these are the organs that, although they are very sterile at birth, they get established very soon after breastfeeding within 5 to 12 hours. And the most important commensals here, please do not panic. These names will become very familiar to you later. So the most important ones that I need you to remember is the first few ones. So Streptococcus viridans is actually a group of bacteria that is found in almost every one of us. And when we do these experiments in the previous years or every semester with the students, and we screen for this bacteria, we from the oral cavity we get positive Streptococcus viridans for almost everybody. The second most common is the coagulase negative Staphylococcus, and this is going to be you're going to learn more in depth in the next lecture. What do you mean by coagulase negative Staphylococcus? So it basically means other staphylococci other than the Staphylococcus aureus, which is the only coagulase positive Staphylococcus. And we have also a lot of Neisseria species and Streptococcus pneumoniae. But please remember that these are the group of commensals, and on the other side we have a group of pathogens. So pathogens means bacteria that are found in some individuals, not all, and in certain conditions, under certain circumstances, they become pathogenic. One of them is Streptococcus pyogenes, and it's a major cause of sore throat or pharyngitis. And again, you will learn about this in the coming lecture. The second one is Clostridium diphtherium, and it causes an infection known as diphtheria. And there is a third one called Bordetella pertussis, which causes a condition called whooping cough. And luckily, in the immunization schedule, we have been immunized against almost C. diphtheria in all of us and Bordetella pertussis, and which we can say that it's 100% does not exist anymore. To the contrary, Streptococcus pyogenes, because we are not immune against it, we still develop, and you will learn in the coming lecture why we are still getting a lot of Streptococcus pyogenes pharyngitis. Nasal cavity, on the other hand, will have two very common organisms that we mentioned earlier, the staph and strep. So Staph aureus and Streptococcus pneumoniae, are very common in the nasal cavity, but again, as I said, they are not found in every individual and they are very likely to be pathogenic. Now let us move on to the growth conditions that are required for these bacteria. So let's say that I took a specimen from the throat of one person. So where do I grow it? I grow it in a media called blood agar because it's an enriched media that will allow the growth of these bacteria. Now let us move on to the eye. So in the eyes again, although eyes we might presume that it's very sterile, but actually the eyes can have also a lot of bacteria, which is fine because our eyes have protective mechanisms. By blinking, we are going to remove any bacteria because of the mechanical movement of the eye very frequently. And also we have what we call lacrimal secretions. So lacrimal secretions, we mean the tears. So the lacrimal glands in the eyes will secrete a lot of lysozymes, which are degradation enzymes that will break down any harmful substances and bacteria. The most important commensals in the eyes are coagulase negative staphylococci. And on the pathogen side, we have Chlamydia trachomatis, Staphylococcus aureus, and Pseudomonas aeruginosa. Please, again, these are going to be mentioned a lot in the coming lectures, and you are going to learn them into details. So do not worry about the names. Now, please remember that we are going to have a lot of cases in the tutorial next week that you are going to understand this topic in a better way and how is it relevant for us as biomedical scientists and also as doctors. Now let us move on to the ear. So we have a lot of also again protective mechanisms in the ear that protects us from having infections. So any opening to the outside environment will have a tendency to, acquire a lot of pathogens from the environment. Luckily, in the ear, we have a lot of wax that will trap and also throw away any harmful bacteria because it gets stuck in that wax. And please, whenever you try to clean your ears without cotton pads, be very careful not to poke your ears because what you're going to do now is going to introduce that wax inside instead of taking it outside. Our ears are actually designed to clean itself. So once you see that wax coming out, you can just wipe it with a tissue or the cotton pad without trying to force it inside. However, we still have some bacteria that can grow in the ear without causing any harm. Again, it's either strep or staph, so it's not very difficult to remember. It's staphylococcus and streptococci. So staphylococci and alpha hemolytic strep. Again, you are going to learn the alpha hemolysis in more details in the gram-positive lecture. However, there are also some pathogens in the outer ear, like Pseudomonas species, and it's one of the common organisms that is found in swimming pools. That's why they chlorinate these swimming pools. They add a lot of chlorine because they want to get rid of all these harmful bacteria. So if you are swimming regularly in swimming pools and you get ears, water trapped in your ears, and if this water is not sterile enough, you might get a pathogen called Pseudomonas aeruginosa or Pseudomonas species. In the middle ear, we have also other harmful bacteria like Streptococcus pneumonia and Haemophilus influenzae. And as you can see from the names itself, these are pathogens that can cause a lot of infections in the upper respiratory tract. But again, these can also be causes of other diseases that we will learn throughout the course. So now let us move on to the lower respiratory tract. So I don't know whether you remember when we mentioned the sterile body sites, we mentioned the lungs. So lower respiratory tract, we have trachea, bronchi, and alveoli. So the lower part of the alveoli and also the bronchi, they are supposed to be sterile because they have a very strong immune system. There is what we call the action of the ciliated epithelium. So it's very efficient in doing an upward sweeping movement, and any mucus will be able to trap any pathogens. And the most important pathogens that can actually be grown from the lower respiratory tract is Streptococcus pneumonia and Haemophilus influenzae. These are the same pathogens that we learned in the previous slide as well. And when we want to grow these pathogens, we usually choose chocolate agar or blood agar, which are enriched media. So please remember this picture here. That actually, I remember I had one of the students asking me about how does this happen, the coughing and the sore throat. So this maybe actually partly can answer your question. So what we have here is a defense mechanism that starts from the mucosa, will trap any harmful bacteria, and then the ciliated movements will move upwards any dirt or bacteria, and then it stimulates the reflux by cough and sneeze that will cause repulsion of these harmful microbes to the outer environment. And again, we have another swallowing like mechanisms, which is a protective mechanism. And actually, that's what we see in children. If they have upper respiratory tract, there's a lot of mucus. They don't usually cough it out. They swallow it because it's a protection mechanism that is easier for them. And once we grow up even older, we become more able to cough and sneeze and expel any harmful microbes to the outside. Now let's just move to the human gut. So I remember mentioning to you that we have a lot of gut microbiome that are very important. So in the gut, we have a lot of barriers that will protect our intestines from harmful microorganisms. And one of them is the acid in our gut. So it has a very low pH. As you all know, acid means it has a lot of like 2 to 3 pH. The saliva that we swallow also is having a lot of proteolytic enzymes that will degrade any harmful bacteria. And also the bile that is made in the liver and then stored in the gallbladder has a lot of bile salts, and these bile salts have a lot of inhibitory actions to the bacteria. Actually, in the immune system that we have also in the gut is very well advanced, and you are going to learn more about immune system in the gut that will actually uh, is very active in fighting a lot of these bacteria. Again, we have a lot of attachment sites in the intestines. So once bacteria get a place to attach, they can survive in the intestinal wall, and a lot of bacteria actually have this preference of varied diets. So what we encourage people to do always is that they should be having varied diet. So what is the concept of having varied diet is that you feed the good bacteria, so you do not uh, cause them to be nutrient deficient. And once you feed them, they will be able to outgrow the number of the harmful bacteria. So why do we need a lot of good bacteria? Because as I mentioned before, we need them to do the task of digestion, vitamin production, and so on. Now, the gastrointestinal tract is actually starts from the esophagus. It includes stomach, jejunum, ileum, colon, and rectum. And these are usually sterile at birth. And once the baby starts breastfeeding, commensals also get established very soon, within 4 to 12 hours. This picture actually summarizes a lot of things, and it's very important for you to understand is that as we go downwards, the acidity of the gastrointestinal tract actually goes up. So from stomach, we have very low pH because of the hydrochloric acid. As you go down, the pH starts going up. So it's in the small intestines it's 4 to 5, in the large intestines it's 7. So this also can explain to you why in the stomach we do not almost have any bacteria, but as you go down you start seeing a lot of bacteria. So in the small intestines you have lactobacilli and so on. In the large intestines we have actually a very large community of harmful microbes. And the commensals here is the anaerobes. It's the most common commensals found in the gastrointestinal tract, and an Enterobacteriaceae. So Enterobacteriaceae are either good and some of them are actually pathogenic. And the most important pathogens in the gastrointestinal tract is Vibrio species, or Vibrio cholera in particular, causing a disease known as cholera, Salmonella, and Shigella species. And there is a very specific growth requirement for these to be selected for, and we call this selective media XLD, and you will learn more about XLD in the practical. So do you think that we do not have any bacteria in the stomach? I need you to think about it and also give me an answer in the tutorial. And also we do have a case about it in the coming tutorial. Now, what happens when you give a lot of antibiotics to a person who needs it? Actually, antibiotics are very good because they are a good invention because they have helped us a lot for the survival of human beings throughout history. It killed a lot of harmful bacteria. But the problem with antibiotics is that it does not select for the harmful microbes. So it will kill even the good ones, or the natural or normal flora. And what happens is that sometimes if you kill the good bacteria, it will outgrow the yeast as well. So yeast is a very dangerous and also harmful type of growth that will happen in the gut when you overdose or when you take antibiotics for a long time, and it will cause a lot of digestive problems. And this is the term known as gut dysbiosis. So gut dysbiosis basically means the loss of balance between the good and the bad bacteria, and that's when the symptoms start to appear. And this happens to a group of patients that receive long course of antibiotics, especially intravenous antibiotics, when they stay in the hospital for a very long time. And please read the notes for this slide. I have written some description of Clostridium difficile infection and how does it happen. And this is going to be also very important for us to understand one of the cases in the tutorial. So please go through the notes for this slide. Now, what happens also when this gut dysbiosis happens, there is an emerging theory saying that a lot of current disease conditions is actually linked to giving antibiotics at very early age during childhood. A lot of children today are obese. A lot of children have autoimmune diseases like and allergic conditions like asthma, diabetes, allergies on the skin like eczema, and also a lot of them have inflammatory bowel disease. And some of them actually will start having these symptoms not as early as child, but again, these can be appearing later in adulthood. Now let us move to the last part of this lecture, which is the urogenital tracts. So here, the urinary system have a very unique system because it has this fluid flow or the urine flow, and urine is usually sterile, and it has urea and other chemicals that will keep the urinary tract clean. Urine actually helps in flushing out any microbes to the outside, and that's why we always encourage people to stay hydrated because hydration helps flushing out microbes as well as any toxins from the body. Because you know that urine is actually one of the major ways of excretion of toxins and chemicals to the outside. So sometimes if you disturb this environment in the hospital by introducing a catheter, so I hope that you know what we mean by a catheter. So it's a tube that you sometimes have to put inside the urethra of some patients to help them pass urine. Some of these patients are handicapped or cannot move. So they need this catheter. Sometimes after going through a major operation, you need to measure the amount of urine or the urine output and you calculate that, so you need to put a catheter. Unfortunately, when you do that, you are very likely going to introduce harmful microbes from the outside to the inside, and this what introduces bacteria to the bladder, causing urinary tract infection. In females, the vagina has very low pH, and that is mainly because of the good bacteria called lactobacillus. It secretes an acid called lactic acid, and also it secretes hydrogen peroxide, which is a known antiseptic, or it's a natural antiseptic that keeps the urinary and the genital tract also clean. So the anatomical site for the urinary tract is the kidneys, ureter, and bladder. Usually we expect all these parts to be sterile, but the urethra is exposed to the outside environment. So the distal one-third of the urethra usually have some commensals without causing urinary tract infection. And most important commensals that we find in the urethra are the Enterobacteriaceae. Do you remember where we mentioned Enterobacteriaceae? In the gastrointestinal tract. So any microbes that can escape from the colon going to the urethra, then it can be problematic. Some of them can remain in very low amounts, so that is not harmful. But some coagulase negative staphylococci, streptococci, and anaerobes, again from the gut, can escape to the urethra and they become pathogenic. If they become pathogenic, it can cause bladder infection or cystitis, and the most important ones is E. coli, Klebsiella pneumoniae, Staphylococcus staphylococcus, and Pseudomonas aeruginosa. You will remember these four organisms by heart because these are also the most common causes of urinary tract infection. And the media that we use to grow urine organisms is called clet. Now, as mentioned before, all parts of the genital tract also should be sterile except the vagina in females, which has lactobacilli. However, there are some pathogens that can cause sexually transmitted diseases like Neisseria gonorrhea, Chlamydia trachomatis, and Treponema pallidum. Please, please remember these names because we are going to study them in details later. So lactobacillus, because they secrete this lactic acid, it keeps the pH of the urine very low, and also the the pH of the vagina very low, and it keeps the other species also in lower amount. If this lactobacillus decrease, let's say, the antibiotic use, it will cause the outgrowth of a yeast that is found in the urinary tract. We call it Candida albicans, and it's a common cause of vaginitis, which is an inflammatory condition or infectious condition in the female genitals. Another co-infection with Gardnerella vaginalis, which is again found in the genital tract of females, and if it grows in large amount, it causes bacterial vaginosis. So, a very quick thought here is that you have to remember that we are all sterile at birth, and we start establishing the flora once we are born within few hours and days. And what determines what type of flora we have is actually three main factors. The first one is the mode of delivery. So if you are born vaginally, it's different than if you were born with cesarean section. And also the type of milk that your mother gave you when you were born. So let's say you were breastfed, then you have a different composition of microbes than if you were formula fed or formula milk. And the third factor, and it's the most important factor, when you become an adult is your diet. So whatever you eat is actually going to determine what type of flora you will have in your gut and in your skin as well. So what we need to remember here, there is a germ theory, but now also there is a hygiene theory. So how many do we really need out of these antimicrobial soaps? It's now believed that overexposure to hand sanitizers and also detergents will likely to kill a lot of germs that are useful for us. And then we need a lot of exposure to germs to revive the normal flora again, and this what actually causes overuse of antimicrobial agents causes the superbugs or the super microbes to grow and causes future problems. So it becomes very important. And remember one more thing is that you have to take care of second genome or your other genome, which is the gut microbiome, and this is determined by the prebiotics. So there is two terms here: probiotics, which is the application of microbial culture we take orally by capsules, and a lot of them actually are lactobacillus, which typically secretes a lot of lactic acid, and we ingest them also naturally from yogurt and from some fermented foods. And prebiotics actually refers to the food that this bacteria will need to eat. So there is either you say you are what you eat or you are what your bacteria eats. So it's very important for you to remember. So I leave you with that note. Thank you. If you have any questions, please let me know. Thank you very much for listening.